they allow potential opportunities to do other stuff. So, and now that was all basically about cornea. I'll share another story that I started with during my postdoc in the University of Toronto in collaboration with some stem cell biologists, and also at the stem cell biologist in the UHN hospital. So this is related to liver, and particularly in liver, basically we have these tube-like structures called bile ducts, and these bile ducts collect all the bile acids that are produced by liver cells or hepatocytes when they are performing the food digestion, and then they transport these bile acids away. Now, the problem is that sometime when for example, the babies are developing, they don't have these cholangiocyte-based bile acids. Or even during adulthood, if these bile acid or bile ducts are damaged, this can cause basically a number of diseases, eventually leading to liver transplantation. And there are so many patients already on liver transplantation list that the waiting list always continue to grow. So our goal was that can we sort of try to create these bile ducts in lab inside biomaterials, and then we can study what kind of diseases or how diseases can be targeted with different basically drugs for these cholangiopathy diseases. Now, our collaborators had developed a protocol by using stem cells and then differentiated these stem cells towards these cholangiocyte cells that can eventually form these structures. But during that process, they had used these mouse cells, which are very difficult to go through clinical translation. So we thought that maybe by using biomaterial engineering approaches, we can actually remove or replace this mouse stromal layer with something that is more well-defined so that it's easy for it to go through clinical translation processes. Okay. So again, I'll skip a lot of chemistry here. So what we did basically, we developed a hydrogel. We sort of created or conjugated signaling molecules on this hydrogel film. And then the idea was that we can control the activation of that signaling pathway that is really required for the differentiation of bile duct cells in a spatial manner by only shining light in different regions where we want the cells to get activated. For example, this proof of concept study showed that within the same well, we are able to nicely create or activate signaling pathway in only part of the cells, while also keeping the signaling pathway sort of off or inactive in the other part of the hydrogel by using a photomask that only allows UV light to pass through in certain regions. So this showed that we can very nicely control where and when the signaling pathway gets activated. We also showed that actually, for example, we can activate the signaling pathway on day 5 to differentiate the cells, but if we don't shine light, then the hydrogels do not differentiate, and that jagged one sort of protein stays off. Okay. We further modified this hydrogel to grow the cells inside this hydrogel, right? Our body is 3D. If we are just growing cells on top of a surface, that does not really mimic what happens in a 3D environment. So we wanted to put the hydrogel, the cells inside the hydrogels to see how basically we can improve their growth. This actually shows very nicely the effect of a protein called laminin inside the hydrogel, where all these basically dots, these are cells, and initially these are not really growing if we don't include a natural protein called laminin. But as we increase the amount of laminin in the hydrogel, we start to see the growth of these cells into these beautiful cyst-like structures with a hollow center. And these are actually cell nuclei in blue aligned very nicely into this tubular shape. Okay. Again, I'll skip this. Basically, one of the goal in the lab is to evaluate how mechanical signals affect cells, and we're focused on how viscoelasticity is affecting. So we want to identify which pathways are involved in evaluating or not evaluating, sort of sensing the viscoelasticity of the matrix. We showed that actually YAP signaling is a key molecule that is involved here. So I'm going to skip quickly this. So, okay, this is what I want to really highlight here. So when we put cells as single cells inside the hydrogel, And the hydrogel contains this protein called jagged1 to activate signaling. We showed that if the hydrogel has appropriate biomechanical properties, which is basically the fast stress relaxation rate, the cells are able to grow into these beautiful cord-like or tube-like structures. And by day 14, we can already see that the cells went all the way from single cells to forming these branched network of tubes inside the hydrogel environment. And if you zoom it down, you can see that these tubes are hollow. They indeed are tubes where the nuclei are aligned here and the center is hollow. This is a 3D confocal imaging of this branched network of tubes where the tubes are connected with each other to really show that this is happening in a three-dimensional space. But on the other hand, if we provide that signals, but the hydrogel is a slow stress relaxing hydrogel or a rather stiff hydrogel, the cells sort of stay as single cells and do not grow. It shows the power of mechanical signals, not just the protein-based signals, which motivate cells or stimulate cells to grow and form these self-organized structures. Okay, so what we have done so far is that we have created these bile duct structures. But remember, I started with liver, right? And liver is much more than bile ducts. A liver contains a network of obviously bile ducts, which takes the bile acids and stores to gallbladder. But liver also is a very big organ. It basically contains hepatic vein where blood flows, hepatic arteries, and it has a lot of hepatocytes. It's a very complicated structure. So we are very fortunate that we are actually one of the very few labs in the USA, probably less than 10 labs, who have been invited for a $25 million project actually by ARPA-H to basically propose the development of an artificial liver in the lab that can be transplanted. What we are focusing on is creating a vascular network that can be bioprinted and sort of mimics that hepatic vein and hepatic artery structure. And then within the wide spaces between these bioprinted blood vessel structures, we aim to embed spheroids or micro tissues of livers that already contains the bile duct structures. So this way, it's a more like a hierarchical structure which contains these bile duct structures, also contains bioprinted these blood vessel structures to basically keep these cells surviving as we increase the size of the liver. And eventually the goal is to create this structure, test this in mice, eventually test this in large animal models, and then potentially for clinical translation in patients. I'll take just a few minutes quickly. We recently actually created a new type of hydrogel that we will be using in the development of liver and also in the stroke regeneration studies. This is also based on the gelatin diethylene hydrogel. One of the key things we focused here is that if we have this hydrogel, it basically can be cross-linked by using the light. The key thing here is that it does not require any photoinitiator. Generally, a lot of these bioinks used for bioprinting or photocross-linkable hydrogels rely on the photoinitiator, which can have toxic effects on the cells. But this is particularly special formulation in a sense that it has this diethylene ring, and when we shine light, it cleaves that disulfide bond, which can then link with another basically thiol on a nearby molecule, creating this cross-link. And in our paper, we recently showed that we can actually use this kind of biomaterial as a bioprinting ink. We can also use it to conjugate different biomolecules. We can also use it as an adhesive for tissue adhesion properties. And we can also basically tune the stress relaxation rate to be able to create a more robust environment for cell growth in a 3D cell culture. For example, I'll show here that when we shine light, if you notice in this region, this dotted line indicates the gelation point, and the flip over of this red and blue curves, this indicates that the sample has gone from a solution state to a gel state. So as soon as we flip on the light, you see that these curves cross over, which indicates the hydrogel formation. So everything happens within seconds, that the solution, the sample goes from a hydrogel or liquid state to a gel state within seconds. And we tested this at different concentrations, and definitely at all different tested concentrations, the hydrogel forms pretty quickly. We can also combine this with physical gelation. Basically, we can incubate the solution first to increase the mechanical properties. For example, it can create physical crosslinks, then we hold it at temperature and then further shine light to improve the mechanical strength. As indeed, just like the Gelma Plus I previously described, the Gel DT Plus also has significantly higher strength compared to a regular 10% Gel DT hydrogel. Again, as I mentioned, that because it has thiols, it can bind to tissue cysteine structure or cysteine amino residues. So it has very nice adhesion. For example, here it's shown with the bare finger, with the rabbit cornea here. We also measured quantitatively this adhesion and showed that this adhesion is similar to fibrin glue, which is clinically used in clinics. We applied this hydrogel as a glue on a wounded cornea as a proof of concept to show that we can use this hydrogel for corneal wound healing applications. It's basically, think of this as if you have a sharp object that suddenly hits your eye and damages your cornea, right? If you do not treat it, it will lead to a scar formation. You will see a white speck basically forming in cornea, which obviously obstructs your vision and also aesthetically is not pleasant to watch. So we wanted to develop a material that can be applied on a wounded cornea so that it reduces the scar formation. So this is basically my size in which we applied the Gel DT, and we showed that the clinically used fibrin glue and Gel DT performs pretty much similarly, where they both can reduce the fibrosis. But because Gel DT is highly tunable, so we think that this is a better matrix to further improve the wound healing processes, because fibrin glue is not a highly tunable matrix. We showed that we can also basically conjugate proteins and release them over time by leveraging the thiodisulfide bonding in the hydrogels. In last couple of slides, I'll quickly mention basically a new type of biomaterial that we have been working on is based on a polysaccharide or naturally derived polysaccharide called fucoidan. Fucoidan is we think is a wonderful material because it can naturally reduce the inflammation at the target site. We've tested this in mice, and we can also basically have shown that it can reduce the antioxidation, or it has antioxidant properties as well. We have created a range of hydrogels starting from fucoidan, purifying it, and then basically creating hydrogels by using different types of cross-linking. We have seen that we can make these hydrogels injectable. For example, this curve shows the force of injection required to inject these hydrogels through a needle. This is important to measure because remember, in the case of stroke, for example, we want to inject the hydrogel in the brain cavity after stroke. So we do not want to apply a lot of the force, because if we are putting cells in the hydrogel, the cells may not like the force of injection. So we wanted to analyze how much force is required when we are injecting this hydrogel. So far it looks promising. It seems that we are able to control the force of injection by tuning the concentration of the hydrogel properties. Recently, we showed that actually we can, for example, when we grow immune cells, we can see that the hydrogel has this natural ability to reduce the inflammation and make the environment more pro-regenerative. My student has been able to show that we can actually differentiate neurons on this biomaterial, which basically makes it a promising biomaterial to be able to study in the stroke environment. Okay, so I'll stop here. What I've been able to show so far is I presented a story on cornea, that how we are using biomaterials to improve corneal regeneration, and then followed by a story on how we are developing liver-based tissue. So currently, as I mentioned at the start, my lab is mainly interested in biomaterial engineering with a clear vision that we want to solve problems. And the problems are related to these tissue regeneration. So we want to create these engineered tissues by analyzing cell biomaterial interactions and then using this knowledge to improve ocular and brain tissue repair and to develop engineered liver tissues for transplantation. But what does the future hold? Where are we going with this? You know, I always think about this. So we have biomaterials. We inject it, let's say, in the stroke, for example, here. And then we analyze this after, let's say, 1 week or 2 week. And for that time period, we have no idea literally what's going on. And to make this process more efficient, I think we need to be able to integrate different technologies so that we are better able to analyze what's really going on after we inject biomaterial. One of the things is just better imaging techniques. Can we image biomaterial degradation rate inside the body in a real-time manner? For example, if we want to regenerate stroke, can we have some real-time or biosensing methods that can tell us about tissue functions? Is brain really regenerating? Can we pick some marker and analyze it sort of non-invasively to see sort of to see that what's happening in that region? Can we tell something about inflammation? Is inflammation going down? When does it go down? For example, if we have a discrete time points of day 7 and day 14 and day 28, we really lose a lot of the information. So I think biosensing techniques can really help in some of these problems to answer some questions that current technologies are simply not able to answer. We can also definitely leverage machine learning technologies and image analysis technologies because sometimes we generate a lot of data and we want to see the patterns that are coming out of that data. So by integrating these advanced technologies, I think we should be able to better develop engineered tissues in future. So I'll stop here. I'll thank obviously a lot of this work, particularly in the later half of the presentation was done by my students. Would like to thank definitely my collaborators as well in Singapore, in Canada, in USA, who have been instrumental in some of these projects, and my funding sources. I'll be happy to answer any questions you might have. So thank you.